Hello, 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 everybody. Welcome to the stream. Welcome, welcome. Hello, hello. Z is just going to be a couple more seconds till she gets in the chat. But before she does, let me say hello to everybody. Hello, Niao. Hello, Robert. Hello, Mylan. Welcome, welcome. Hello, Toxo, of course. Hello, Astral. Welcome, welcome. Hello, Gluon. Welcome. Robert, I already said hi to you. Yeah, yes, I did. Hello, Upstander. Welcome, welcome. And Brenda and Pamela. Welcome, welcome. Hello, welcome. And Dreamless Sleep, welcome. Hello, Marlon. Today, we are going to be talking about the wonderful Tardigrade. And as I learned from Z and Mylan's debut stream, Z has a phobia for the Tardigrade. And of course, having made a whole video about the Tardigrade myself, I cannot allow that to stand. Hello, Leo. Welcome, welcome. What would one make one scared of the tardigrade? That's an excellent question, and I hope when we hear from Z that she will be able to explain herself. I gotta say the real tardigrade mouths are a little 5-om kissy face, but I did not consent to the kissy. I mean, they're not up in your grill like that, you know. They're eternal kissy face. Aren't there lots of other funky microorganisms too? Yeah, totally are. But I could see it. Like, I understand maybe what is a little terrifying about them. The little mouth is kind of void adjacent, right? It's kind of like looking into a mouth part that shouldn't exist like that. They're also awfully wrinkly, but I think they lie in the sort of too, like, what is it? Like, so ugly they're cute kind of space. I certainly think they are, if you ignore the face, very, very cute. Tardigrade mouths look like those vacuum cleaner bag adapters. They do. Their little feetsies are so cute, though. That's true, though, Gail. I actually do agree with that. Although, judging from this EM, I'm struggling to see exactly what the structure of their feet are. If, at this microscopic scale, if there even is, like, a foot equivalent, right? Because they kind of look like AI generated tendrils or something. You don't really have, like, what we would consider to be a normal amount of toes, right? But this image is taken through electron microscopy, which is why you can get such high definition details on all the tardigrade and all the, the wrinkly glory that is the tardigrade. Cute feetsies with void mouth. Yeah. I could see that. But one could argue that this emoji itself is a little bit of a void mouth too. Ooh, Stream Elements bot. Welcome, welcome. Finally in the chat. Yeah, you're good. I think Z is ready here. All right, let me go ahead and unmute the audio and welcome in our guests. All right. Desktop audio is unmuted. Welcome, Z. Hello, hello. Please introduce yourself.
Speeding at C, it's me, Z. Hello, everybody. I am a physics VTuber. I do physics, astrophysics, engineering, aerospace, apparently also export control content. And I'm so excited to finally be collabing. Well, first I can briefly do attendance. So class, if you're here, put your attendance into the chat. Hello, PogChamps up by. Hello, N30W. Hello, Warpy. Hello, Architels. Hello, Astro Choppers. Hello, Eggy. Hello, Brenda. Hello, oh, I already said that. Hello, Rongermonk. Hello, Xenotime. Oh, shoot. I'm muted on my end. Hello, okay.
Oh, classic. Classic.
Well, oh shoot, the tardigrades turned off my mic. They've been sabotaging this stream. They killed OBS and now they killed my mic.
Yeah, my chat is board witness. You did say hi to everybody in your chat.
Yes. So if you are dual watching, then I said hi to them. Well, speaking at C, it's me, Z. Hello everybody. Wow, this collab has been in the works for quite some time, actually.
Yeah, we've been trying to collab since like January, and we've been cooking up a couple of different ideas, but every time it came to actually doing the collab, something or another would cause the collab date to shift or get canceled or something. But I'm kind of glad it did, because we went through a bunch of ideas that were maybe not as exciting as the current topic today, which are tardigrades, which I truly, truly love dearly. But Z, my chat is asking, to get us started, what's up with you and tardigrades?
Oh my god. Oh, the story is crazy. So, when I was very little, I would read these like National Geographic magazines. Um, and I remember one day one came in the mail, and on the cover was this humongous tardigrade, and I was terrified. Um, and since then, it just looks weird. Even when we were setting up this stream, I remember, like, you were like, "Oh, I'm gonna screen share now," and I was like, "Okay," and I get jump scared by this creature. Like, it just looks weird. It has claws, like, look at its mouth, like, it looks really, like, odd.
You don't think my little Photoshop job made it any more appetizing, any more appealing?
I mean, like, if you put a little bit of blush on a creature that looks like this, I mean, it kinda neutralizes, like, halfway there, but
Yeah, that's all I can really ask for.
ooh.
Uh, all right. And for Z's audience, hi, I'm Phi the Neutrophil, your cell and molecular biology VTuber extraordinaire, and I do a lot of pathogen stuff. But today we are not talking about a pathogen. We are talking about instead a very healing and peaceful microorganism, the tardigrade. And I promise Z, this is only one of two actual pictures of a real tardigrade because I figured if my goal here is to convince you, Z, that they are worth loving, I'm gonna try not to show too many real pictures of them.
Okay,
uh,
good. Because as a visual learner, this picture already freaks me out. Like, I've been looking at it and, like, trying to remove it from my brain.
all right, so let's get off this screen and let's just get started, shall we? So
Mhm.
yeah, in order, I think, to have you be rizzed by the tardigrade, I think we have to talk a common language. And the thing that biologists and physicists have in common is this idea that we both are at least differently fluent in the world of molecules, right? So, again, I'm not going to show you too many pictures of tardigrades because I know you find them unpleasant, but maybe let's focus on the molecules that make the tardigrade. And I think from there, you may come to appreciate the existence of the tardigrade from a molecular perspective. Does that sound good?
Yeah, I love this meme, by the way. Look at how strong that physicist looks. Like, ooh.
This is a friendship forged in fire, and we will see a little
Yeah.
bit of that in the upcoming slides. So, if you did not know, I made a whole video about the tardigrades' molecular mastery over the elements, which you can find in my video description down below if you're on my channel. I had this idea for this video where I wanted to talk about tardigrades because I knew that the internet loved tardigrades, but I wanted to do so not from like a zoology perspective because I don't know anything about animals, but about their molecules. And so if you're curious, if this stream has made you curious as to what those molecular mysteries are, you can find that video in my description. But for this stream, I will be recapping some of that information, updating that information, and also sharing some new things that I researched for this collab. All right. So I wanted to connect with you here, Z, right? Because you are our resident space VTuber. And I don't know if you know this, but tardigrades are actually capable of not thriving in space, but they are surprisingly resistant to the hostile environment in space, right? So tardigrades are extremely cold resistant, able to survive at temperatures as low as minus 270 Celsius. They can survive for a short period of time the vacuum pressure of space, the myriad ionizing radiation you find in space, and oxygenless environments, and they can survive being dried out. So I just wanted to preface this by saying, as a space VTuber, I strongly believe we have a case here for you to enjoy the tardigrade. And up here is, of course, Toxo's wonderful art of the tardigrade, hopefully a little more appetizing to you than the EM images.
Yeah, no, I like this little, like, cute little doodle. Um, I have to admit, like, tardigrades are really resilient. I don't know if you know this, but I actually heard recently that there was a bit of controversy because, there was a space mission that apparently happened where this mission took a bunch of tardigrades to the moon. And because tardigrades are so resilient, like, this is actually, like, a direct sort of threat to the natural environment of the moon because these things can probably even survive on the moon. Is that correct?
So what I'll say is that they are not known to survive for very long periods of time in space. I'll actually get into the definition of this in the next title slide, which is they are extremotolerant. So the difference between a tardigrade and something like archaea that live in a hot spring is that tardigrades are tolerant of extreme conditions. They do not thrive in them, right? So that's a really important point to get across immediately. So it's not like they're chilling in space, rather they are braving space. That's a more accurate description. They are yeah, so I wouldn't really be too worried about tardigrade colonization of the moon, considering that if they are in the moon and they are exposed to that vacuum, they are probably curled up in what is known as a ton state, which we will get into very shortly. But before I get into the body mechanics of the tardigrade, given what we know about the tardigrade's extremotolerant nature, I wanted to give both our audiences a little bit of time to think about evolution, right? Evolution is sort of the unifying central principle in all of biology, which essentially states that the environment, the ecology of an organism dictates sort of how it evolves, right? And so, as we've seen in this previous slide, this tardigrade is capable of surviving many, many conditions that are very rarely found on planet Earth. And so, I think an interesting question to ponder is what unholy, hellish environment could the tardigrade have evolved in to make them so resistant? And this is a question that I'll pose to you too, Z. So just to give our audience a bit of time to think, and yeah, I just kind of want to get your thoughts on this, Z. Like where do you think the tardigrade normally lives where they would be able to evolve such extreme resistances?
I mean, first off, that little doodle is so cute.
It's good, isn't it? Toxosaur is so good.
I wish tardigrades actually looked like that with like hearts on their feet.
You caught it. That's the classic Toxo touch there.
I think don't they live in like hot springs?
They can be found in hot springs, yes.
Maybe I don't know if that's where they evolved from, but I do remember that they can live in like Yellowstone hot springs and stuff. Um I'm pretty sure they also live in water, but I feel like that's not very unholy.
Okay,
I think
water, you're getting
water's
closer,
quite
Z.
holy.
Water
Water,
is part of the correct answer here. Yeah, I don't think anybody will actually get this if you didn't know beforehand or if you didn't watch my video, but it's like good to give this question a little bit of time to appreciate the answer. And Z, you've got half the question right. This is kind of an aquatic environment, but not how we traditionally think of aquatic environments, right? So it's not a lake or a river or a pond or an ocean, but water is involved here. So we'll proceed on, but keep that in mind, Z, that you did get it
well, chat
half
says
correct.
very hot water or in the early Earth ocean. Yeah, no, that sounds about right to me, but
Yeah,
I'm not
yeah.
sure.
And my chat is saying deep thermal vents or geological conditions, all excellent guesses, but actually the tardigrade's preferred environment is a lot more pedestrian than all of these extreme environments. A has got it. Moss or sometimes just on little water droplets on leaves. And this, to me, was really surprising. So you can often find tardigrades hanging out wherever there is water. That is their preferred environment. So even though they are capable of surviving things like hot springs, where they are normally found are aquatic patches in like foliage, right? In grass, in leaves, and as Aya said in my chat, in moss. And so that is really surprising because you don't typically think of this environment as being all that hostile to life, right? This seems like a pretty easy place to live. But that is because on the space on the leaf where there is water, that's a pretty okay place to be. But what happens on the patches of leaf where there is no water, right? We have to think about the micro scale, right? So on a leaf where there is no water present, what is an animal supposed to do when there's no water around? A lot of animals would just die, right, in absence of water. So actually, a lot of the tardigrade's resistance can be attributed to evolving a way to counteract the instability of the presence of water, because you might have water, you might not have.
Whoa.
And so what we're going to see is that everything, or not everything, I shouldn't say everything, but a lot of the tardigrade's resistance to freezing, to heat, to the vacuum of space, that all stems from this evolutionary need to not die when being dried out. And I think that's a pretty interesting superhero origin, right? Like the tardigrade was not forged in fire. The tardigrade was forged just on some modest grass, which I
On a
think is pretty
leaf.
nuts. Yeah, on a leaf.
Wait, so Eggy in my chat is asking if I drink tea, I'm drinking tardigrades. So if I'm drinking water, is there tardigrades in that?
I don't know the exact answer to that, because that would be more of like a sanitation regulation kind of question, which I'm not entirely familiar with. But if you know, have a water sample that you take unfiltered, there's a decent chance that there's maybe a tardigrade in there. I don't know. Just think of Toxo's Don't think of the actual EO. Just think of Toxo's
Wait, my chat also has a question. Are they
Yeah.
related to insects in any way, or I
That's
mean, these are microbes, right?
yeah, they're micro. They live at the microscopic scale. I'm not a zoologist, so I actually don't know who their closest animal neighbors are. That's an excellent question, and I'd love to know the answer if someone in chat wants to quickly Google that.
So they're basically everywhere, right?
Yeah,
That
more or
means.
less. They can survive in all sorts of conditions, but they prefer to
Oh,
be in water. Don't worry,
this is making me feel more
No,
terrified.
no, no, no. We're gonna get there. We're gonna get there. Again. Just think of Tox's cute art. They're little buggers, little hearts for feet.
Ugh.
Okay, we'll go on. We'll go on. So why would desiccation or drying out be so deadly for an animal? Well, there are two main reasons that I can think of. One is this idea of tonicity, right? So your cells are little water bags full of salt, and they are in an environment where there is water and also salts on the outside. And this is an example that is drawn from Wikipedia that describes what happens when there's too much salt on the outside, too little salt on the inside, or just the right amount of salt on the outside, right? So in this middle condition where we see isotonic cells, these are cells that are happy. There's not water leaving and there's water leaving and exiting, but at the same rate, so they retain this healthy blood shape, right? When there
Mhm.
is like very pure water on the outside and not enough salt on the outside, you get the condition known as hypotonicity. And so this forces water into the cell, which can cause the cells to burst. And if the reverse is true, if there's too much salt on the outside, that can actually draw out moisture from cells, causing them to shrivel and die. So hypertonicity is what you experience when you try to make pickles, right? When you salt your vegetables, it draws the water out of the plant cells. The same thing can happen to, mammalian cells or animals, right? So this danger of having water drawn out of your cells is a very significant threat that as humans, you don't really think too much about, but on the microscopic scale, this is life or death. This is extreme, right?
So the tardigrades are basically dealing with this all of the time, right?
Yes, exactly. And the reason why they're dealing with it is not necessarily because of salt levels, but because they either have water or they don't. And that can change, throughout the day, even, right? A particularly hot day, you dry out all the water around, all of a sudden there's no more water, and now your cells are drying out. So a lot of the strategies that the tardigrade has evolved has to do with maintaining survival in those harsh conditions.
Wow.
Yeah.
Well, that's a pretty harsh environment to me. I did not know that, like, an environment that looks as calm as a leaf is basically like a battleground for these tardigrades.
Yeah, that's a really great way to put it. It's a battle for survival because you never know when you're going to get water.
Wow.
Yeah.
Well,
So
I have more respect for the tardigrades now.
so we have another picture of the tardigrade, but this is not as high resolution, so it just kind of looks like a blob here. So hopefully this is a little more doable for you, Z. So
A little.
A little bit, right? Not as much wrinkles, but this is important to see from just a mechanical point of view, and it's something that the tardigrade lovers would kill me for if I didn't talk about. So when the tardigrade goes under stress, it transforms into what is called a ton state. So on the top, you can see an active tardigrade. This is the tardigrade that would be chilling in water, very happy, eating and chilling, relaxing. But when they dry out, they revert to something called a ton state, where they tuck in all of their legs and they form a little croissant looking thing. And so this is called the ton state. And so, talking about colonization of the moon with tardigrades, if you were to introduce tardigrades into the moon environment, they would probably all tuck into this little croissant shape and try to wait out the time that they have to spend on the moon until they are reintroduced to water and all the good stuff that makes life on Earth possible. Is this exposure therapy for Z? Well, I don't know.
How long can a tardigrade stay in this ton state?
Depending on the conditions, it can be for quite some time, although I don't have the number on me at this very moment. But they can remain in here for a very, very long time.
And so, like, is it alive in the ton state? Like, if you just sprinkle some water on it, will it, like, undo it? Or how does it know when it's safe to reemerge?
That's an excellent question, and not one that I am 100% ready to answer, especially because I don't think people know yet. The mechanisms by which tardigrades are capable of surviving the elements is still an active field of research, and we're going to be covering some very recent research, some insights as to how the tardigrade is doing this kind of stuff from a molecular point of view. From a whole organism point of view, I think it's even more mysterious. But to answer your first question, yeah, if you sprinkle a little water on the tardigrade and the conditions are right, they'll come out of hiding. So they just need a little bit of water and they are good to go. All right. So
Wow.
this is the last image of a real tardigrade, and now it's just cartoons from here. So you can take a breath.
But now I can never eat a croissant again.
Oh, the fear goes so deep.
I'll just be thinking about tardigrades in the ton state.
Okay, so let's talk a little bit about the molecule. So yeah, so up until now we've been talking about the organism, but now I want to speak our shared language, our shared understanding of molecules. And so this is a protein that I had covered in my tardigrade video called Cas8, and this is what is known as an intrinsically disordered protein, or as I like to call it, floppy spaghetti, that aggregates in response to being dried out. So if I were to show you this noodle, this coiled region, right? You see this coiled region, this zipper-like region, that is an area where the protein is actually structured. So that part doesn't really change. It doesn't really bend all that much. That part remains pretty strong, right? You can think of it as having like high tensile strength, high whatever physics term you want to put on it. But you see this thin line over here, the thin line at both ends of this long curled noodle. These are what are known as intrinsically disordered regions, or in other words, when you have these proteins in water, those strands of proteins can flop very freely. So they don't retain structure. They're kind of just like loose string. The problem...
Whoa.
Yeah.
So there's one part of the protein that's like rigid and strong, and then there's another protein that responds to the lack of water.
So this,
is that correct?
This is the protein that responds to the lack of water. So we're going to see how it responds in the upcoming slides.
Oh, I see.
Yeah.
Okay.
It's just that there are two parts. You see the thicker ribbon that curls, that like has a very regular helix formation like DNA. It's like the blue colored region. Yeah, that's the region of the protein that doesn't bend, that stays that shape. And then the rest of the protein, the thinner portion, that's kind of like just a jiggly mess, that is free to move in all sorts of different conformations when it is in water.
I see. Okay, well, I guess you'll probably explain like how it responds to desiccation, right?
Yep. And to my audience, this is going to be very, very surprising. So I first want to go over what exactly about drying out makes for a very deadly condition. And I think Marlon in my chat had pointed it out earlier. So desiccation is not only bad because it dries out your cells, but it also causes your proteins to cook. if you could believe it. Not cook
What?
like in a temperature dependent way, but cook as in it destroys proteins. If you remove all the water from a biological sample, there is a very good chance that a lot of those proteins end up destroyed. Now, that's not true for all proteins, but for a lot of proteins, that is true. And so I have here just a simple mock diagram of a made up protein and some water molecules that are surrounding it. Right. So water keeps proteins electrostatically happy in solution. Right. So again, if we're talking about the shared language of molecules, you know that on one end of water, there's a positive end, and on the other end, there is a negative end. So on the red side of the water molecule, there is a slightly negative charge. There are slightly more electrons are spending slightly more. I have to get this right because it's Z, right? I can't half-ass the explanation. There is.
I'm watching carefully.
Yeah, so you can correct me if I'm wrong, but a partial charge refers to the fact that electrons will spend more time on the red side, the oxygen side of the water molecule, which gives that red end of the water a little bit of a negative charge. Not a full negative charge, but a little more negativeness, you could call it. And then on the hydrogen side, which are the blue parts of the molecule, you have a little bit more positiveness. And so it's this polarity, this difference between the sides of the water that allows it to be so good at dissolving proteins, right? So this made up protein is made of some slightly positive amino acids and some slightly negative amino acids in a chain. And you can see that water with their charges is able to keep this protein happy in a water shell.
Yeah, no, water polarity is great. I mean, even beyond like proteins too, it's used to, I mean, it gives the water a little bit of stickiness, right? Um, so that the water can sort of, I wouldn't say like stick together because it won't actually stick together, but it gives like cohesive properties.
Yeah, yeah.
And it's really cool to see the protein, like basically it's different parts of it are interacting with water differently because the amino acids are also slightly positive and slightly negative, and that is attracted to the slightly positive, slightly negative parts of the water.
Exactly, exactly. That's exactly right. See, so the properties of proteins, because we are water-based life forms on Earth, are dependent on the fact that there is water around. If you remove that water, all of a sudden these proteins that have this lovely structure no longer look the same. So when water is removed, the neutral amino acids in the core of the protein, right? So like the charged amino acids, they love water, they're on the outside of the protein. And the neutral amino acids, which are more fatty, you could call them, like oil, they usually stay on the inside, right? Because like dissolves like, very basic principle in chemistry. But if you remove the water, that internal neutral amino acid can now come out to play. And this has disastrous consequences because if you oil likes to dissolve oil, water likes to dissolve water, and if you have this oily amino acid core exposed to the outside, what can happen is that another protein with a neutral amino acid core that's exposed can bind to it. So not only do you get unfolding of these usually pretty structured proteins, but now they're sticking to each other in an aggregate, a mess, a protein mesh, like scrambled eggs, right? Like when you heat up the egg protein, it goes from nice, egg white albumin, but then when you start heating it up, you start to tear apart those proteins, get those neutral amino acids in the center of those proteins to stick to each other in all sorts of disordered, unorderly, random ways, and that's how you get this mush that is a cooked egg. Does that kind of make sense?
When you say the protein is cooked, it's not like, like putting into a fry pan and cooking it, but it's basically like breaking it apart and having it stick in the wrong places.
Yeah, in an irreversible fashion. So chemically, it's somewhat similar to being cooked. It might not look the same, but it's destroyed the proteins. So that is what I am referring to, because there's no coming back from this because it's an aggregate.
Wait, there's a quick question in my chat.
Yeah.
Wouldn't the water break down the protein through hydrolysis?
Not all proteins are susceptible to hydrolysis, and hydrolysis is also typically catalyzed by enzymes. Yeah, I don't think that protein. I don't think. I mean, proteins can hydrolyze spontaneously. I just don't super think that, apart from an enzymatic reaction, that it happens too often spontaneously. That would be my best answer there. I hope that makes sense. Someone in my
Pretty
chat
cool.
says, insert hour-long lecture by my physical chem professor about hydrophobic effect is caused by entropic effects in hydration cages. Yeah, this is my way of
What?
making that accessible. That's a lot of fancy words, but this is my like 101 version of that.
No, this is very accessible. As somebody who, I mean, I didn't suck at chemistry, but I have to say that biology and chemistry is not my strong suit. And this, like, oh, this pink circle is slightly positive is definitely very understandable.
That's good because I also suck at chemistry, so in part I have to do this for myself too, because this is the only way it makes sense to me. So I hope now we can appreciate that drying out on a leaf is like as insane to the tardigrade as like being thrown into a hot spring or being cooled by liquid nitrogen, right? It's as deadly because once you start removing water from a system that has proteins, to borrow technical language in the chat, you remove that hydration shell, then all of your proteins start aggregating, they start getting quote unquote cooked, and then all of your proteins are destroyed. So drying out, very, very, very dangerous. And so if you look at this in an experimental point of view, if you are an aspiring biologist, you might see that when you are working with proteins that are correctly folded and are functional and happy, they just look like water, right? Because you dissolve proteins in water, you don't see anything. But if you remove too much water from your protein sample, what you could see is something called precipitation, right? You get garbage at the bottom of the tube, white film or powder that forms, and that represents your proteins getting quote unquote cooked or destroyed. They no longer are happily dissolved in water in their hydration shells. They're left to rot on the bottom of the tube. And so you can imagine that, in a tardigrade, if you imagine this tube as a tardigrade, it wants to be here where all of their proteins are happily in water. Once you start getting proteins crashing out and denaturing inside of you, that's when things get really, really bad.
I
Very, very
wanted to say,
dangerous.
Fi, this rhetoric is very interesting. The proteins that crash out of the solution, the proteins are cooked. What are these? These are like
So
Gen Z terms.
okay, we were there first. By we, I mean chemists.
The proteins are crashing out.
That's so true. I didn't
What do
even
we
I
do?
didn't even realize that's so true. Yeah, they're crashing out, dude. That's so
Chat,
true.
is the protein cooked?
Oh, dude, you're actually so right. Are Gen Z? Are you all just chemists?
Yeah, the chemists were there first. Yeah, it's just chem lingo, exactly.
Chem lingo. Yeah, Toxic says Gen Z is appropriating chemistry.
Yeah, no, it's cooked.
That's so true. It's cooked, dude. That's so funny. All right. So here's a question. And again, I don't expect anybody to actually figure this out unless you watched my video or know the answer beforehand. But it is, again, an interesting point to just pause so that we can appreciate how incredible the tardigrade is. So I'm telling you that this protein here, right, this rigid protein that's mostly an alpha helix, but it has floppy spaghetti regions. I'm telling you that in order for the tardigrade to survive, this protein begins aggregating. It starts binding to each other. It starts forming these protein structures that are layering on top of one another. And somehow that is helping the tardigrade survive being dried out. And again, yeah, Z, there's very little pressure to get this right because, again, there's no way that any human, I think, could have figured this out. But just for fun, do you want to take a guess? How could protein binding
I don't
stop a
know.
tardigrade from drying out?
Maybe like the little wobbly regions like combine and it like forms a giant wall of the rigid regions.
Okay, I mean, you're thinking in kind of the right direction here. There's something structural about how this protein forms aggregates that is going to be very important for survival with no water. So I'll have you keep that in mind that you said that, right? There's something about these floppy regions that are going to be really important for the structure, the internal structure of a tardigrade, the tardigrade's body, to help it not dry out.
Maybe chat if you guys have any ideas.
Yeah.
People say it traps water. There is a lot of surface area, so lots of area with polar charges. Or the protein acts to forcibly hold parts of other proteins apart that it shouldn't collapse. Lots of
Yeah,
cool ideas.
those are cool ideas. We'll see if they are right when we start talking about the rest of the presentation. Very interesting hypotheses here. I just want to point out a comment in my chat that says, you want to explain hydrolysis to the youngins by saying it's water rising up a molecule, causing it to break up.
What are these verbs?
I think the children will be all right. If that's
Does it
the way they're
provide
learning.
like a hydrophobic layer?
Ooh, a hydrophobic
Ooh.
layer. That might be. Uh, I would have to know a little more about what that commenter meant by hydrophobic layer, and how that would help
Hmm.
being dried out. Because from my impression, a hydrophobic layer would repel water, would kick it out.
Maybe a hydrophilic layer that attracts water.
Yeah, maybe. And I've seen some good guesses in chat too that I don't want to say out loud because spoiler alert, people have, combined between our two audiences, people do have the right answer. It's very, very interesting. Did y'all look it up or watch my
Ooh.
video? I could not have figured it out, so y'all are smarter than I am. Okay. So to defeat protein aggregation, aggregate yourself. So this protein, as I said, forms these aggregates, these tough to destroy protein aggregates that are, in most cases, deadly for an organism, right? I know my chat is very familiar with this idea of aggregating proteins,
What
but
is that?
your audience might not be as familiar. So this is what is called a prion amyloid fibril, and your audience, I don't know how familiar your audience is with neurodegenerative diseases, but there are this class of diseases that happen in the brain, especially in old age, that are tragic, right? They rob you of your memory, your motor function, et cetera, et cetera. And these diseases, the backbone of a lot of these diseases, are the fact that there are these little proteins that do the same kind of aggregating in your neurons. They build up into these cooked protein regions that your cells have a really hard time dealing with. And when they build up over time, can kill neurons. So, classic ones are Alzheimer's disease, which can cause memory loss in old age, which is, again, very tragic. Parkinson's disease, which can affect your motor function, right? Parkinson's patients have somewhat, you could call them debilitating tremors, inability to move their body in ways that they would like to. And Huntington's disease, I know less about, but I think it manifests similarly, where there's behavioral change, motor change, and degradation over time. And these are oftentimes untreatable, at least with modern medicine, although we're starting to see changes with that in the
Oh,
future. These
these are the
yes,
prion stuff.
yes, these are prion diseases. The first three are
Oh
prion-like
God.
diseases because they are not the mammalian prion protein, but they act in a way that is similar to how prions work. And then these four that I highlighted in red, these are true prion diseases. So mad cow disease, if you are boomer enough to remember the mad cow scare, that was very scary, where you could contract a deadly prion disease from eating beef, and it would take years and years and years for symptoms to actually show up, but it was always fatal when it did. Kuru, CJD, fatal familial insomnia, which robs you of your ability to sleep. These are
Oh
really
no.
devastating diseases that all have this principle of aggregating proteins at their core. And so what I'm saying is
Oh,
amazing about the tardigrade is that it has an aggregating protein, but instead of giving the tardigrade an incurable, tragic disease, this aggregation in the tardigrade is actually adaptive. It is something that saves it from being dried out. And I think to set up what makes this so incredible, I wanted to point that out because, again, when I read protein aggregation, my head immediately thinks Alzheimer's, Parkinson's, mad cow, these terrible diseases. But in the tardigrade figured out how to make aggregation protective, and I think that is amazing.
wow. That's really cool, but oh, well, I have to say prions are probably up there in terms of things I'm scared of, including tardigrades. I remember Faye and I did a collab and she was explaining it was for a Halloween stream, I think. We were talking about spooky diseases and prions and mad cow disease. I can't believe the tardigrades have actually sort of used this to their benefit.
Yeah. And it's worth noting that the tardigrade aggregation is reversible. So there's something about the way that the tardigrade protein binds to each other that allows it to be reversed. So kind of like uncooking an egg. And that is really, really a great mystery to solve, right? Because if we can figure out what makes it so that the tardigrade can unfold its aggregates, maybe there's some hope for us for these terrible, aggregation diseases as well. So there's a lot of information that we can glean from the tardigrade that might have relevance for human health, and I think that's encouraging, I'd say.
Are there a lot of studies out there that are using tardigrades to understand how to like undo these proteins?
There are we'll actually talk about medical applications about tardigrades later in the stream, but to answer very briefly, yes.
Wow, that's really cool.
Yeah. Okay, let us go on. So here is a figure directly from the paper that I have linked in the description below, and here we see an increase in Cas8 concentration on a plate. So this is happening in a dish with, some physiological buffer. And A, B, C, D, this is increasing amounts of the tardigrade protein. And I want you, Z, I know you are not a biologist, but it's okay. I think this is accessible enough to take a guess. So first, chat and Z, describe to me what you are seeing as you go from A to D, and what you're actually seeing in the image are fluorescently labeled tardigrade protein, this Cas8 protein, right? So when you see these lines, this is how this protein is folding. And so what do you notice, Z, as you go higher and higher concentration here?
Well, at first there's not that many of them, but they look very like linear. And then in B, there's a lot more of them and they're sort of scattered throughout the surface. And then in C, it kind of looks like cork, I guess. I
Yeah,
mean, I
yeah.
could try zooming in a little bit more. And then in the final one, it looks kind of spongy. There's a lot of holes in it.
Yeah, it's spongy. Like it aggregates in a way where there's space in between the protein, right? So we're going from these fibrils, these like devastating structures that I showed before with the prion aggregate, but with enough of this protein, you actually start to form something like a sponge. Is there a more general term you would use for a sponge, Z? A foam-like, I think that's good.
Oh.
This is a general term for something that could be this, something similar to this. I'm wondering if we can pull out the answer from chat. If anyone has
Something
guesses.
similar to a sponge, like porous?
Yeah. Oh, Lunkel says this looks more like atomic force microscopy than fluorescence. You might be right. You're probably right. There's no. Yeah, actually, you're almost certainly right, because this is at this resolution, it's very unlikely for it to be fluorescence, because you can't actually resolve this deep with just fluorescence. Thank you so much, Lunkel, for catching me on that. It's been a while since I looked at this paper.
Well, my chat is there's like 4 people saying porous and then absorbent.
Mm-hmm. I actually have someone in my chat that's got the word I'm looking for, which is not necessarily one that would come immediately to mind, but it forms a gel, right? So if you were to zoom in on a matrix gel, it would look something like this, right? So like a gel, as in like a broad definition of the word gel, right? Like regular repeating patterns with water
Whoa.
sort of in it, right? So when you think of making jello, right, what is jello? It's gelatin that has been heated up to form what you would describe as a gel, right? And so what we're seeing here is actually a similar phenomenon, right? So we've added enough
Wow.
of this protein to form a gel. And why might a gel actually be helpful to survive desiccation?
Wait, before we go on, I did have a quick question.
Yeah.
So is this kind of porous structure that we're seeing here what we would expect if we saw like, I don't know, like jello under a microscope? Is this like a biological term for gel? Like, is
Uh,
that different?
So, from my understanding, and it's very limited because I'm not like a materials guy, is
Mhm.
that a gel is refers to like a protein matrix that has water incorporated in it. Yeah, that's my best description of what I believe a gel to be, although I'm a little out of my expertise in the actual material science of things.
Yeah.
Also, the
And
authors call it a gel.
I guess this is useful because it kind of I mean, chat says it's like a mesh catching stray water. It captures the water itself, right?
Yeah, that's what the authors of this paper hypothesized. They looked under a microscope and saw that the tardigrade protein was forming this gel-like structure, and they actually. So your chat is very smart. My chat also got it right too. But the hypothesis here is that if it forms a gel, it can trap that water, and that way, when you dry out the tardigrade, it never fully dries out. It can capture that water in some form or another.
Wow.
Very smart, guys. Very, very cool.
Wow. The tardigrade is very smart. Well, how did it create this, like I guess it probably evolved over time to use this protein in the first place, right?
Yeah, so we're actually going to talk about how I think the evolution of the tardigrade happened at the end
Okay.
of the presentation. But that's also like, as a biologist, I look at the tardigrade and I think, I can't help but think like evolution made you. I mean, evolution made all of us, but evolution like really made you had to go through some
Yeah.
stuff to actually be able to come up with a mechanism this clever. Um, so I think that's very exciting. It's also worth noting that this is a pretty small protein, which might make it a little easier to evolve than something that is more structurally complex. So it's
Ooh.
worth noting. Evolution 8 with this one. How many Zoomers are there in chat?
Why are there so many Gen Zers?
Okay, so you already answered why a gel might be useful in terms of drying out, and there were a couple of acceptable answers here. So these are hypotheses, right? So when you form a gel, you reduce the volume of water needed to, to maintain correct structure. The spaghetti parts
Mhm.
of the protein also contain water-like amino acids, and I think either you or somebody in your chat had hypothesized that as well, right? These floppy bits of protein are water-like and could potentially help proteins fold normally, because what does a protein need water for? All a protein needs water for is having something that is a little negative and something that is a little positive. And the floppy bits of this protein offer the same electrostatic interaction as water does. So that is a possibility, right? And then finally, as we've said, this gel structure might prevent water from fully escaping the tardigrade, allowing it to maintain some moisture on the inside.
Wow.
Uh,
Mhm.
However, there's a twist here. Oh, sorry, there's a plug before that. Yeah, so I go into a lot more detail about the mechanism of this in my tardigrade video, and I do like animations and things, and I play with the molecules to try to get an idea for what Cas8 could actually be doing to proteins. So if you want a little more literal imagery, then please go check out my video in my stream
Yep, and
description.
I just linked it into the chat of my stream as well, so go check it out. I
Thank
will
you.
also be watching it right after this stream.
Thank you so much. Okay. So this third idea, the one where most of us I think would go to when I've been leading you all to think that this gel could be trapping water, is also something that I thought was true. There is another paper that asks this question that I did not read when I did that original tardigrade paper. And I have since read it, and let's just go ahead and take a look at that title. So this was published in June 2023. I
Oh,
published the tardigrade
no.
video in December 2023, which means this was actually out before I made my video, but I mean, I'm one guy. I cannot read every tardigrade paper, so I was a little sad that I managed to miss this, but this is a paper that actually tests this hypothesis that the Cas aggregation proteins are keeping water in like a gel. And as you can tell from the paper title, the tardigrade protein interacts with but does not maintain water in hydrated and desiccated systems. So that third hypothesis might not be true, but we're going to see just a little bit as to whether or not how much we buy that, right? So something that I do a lot on my streams is critical analysis of papers. So we're not going to just say, oh, we're wrong, but we're going to take a little bit of a peek as to how wrong this idea potentially is. All right?
Yeah, let's see.
All right. So is this gel thing true is it forming water? First, I want to show this is actually fluorescence. So Lunkel correctly pointed out that that previous image was almost certainly not fluorescence. It might be atomic force microscopy. This is fluorescence. So we are now zooming out. These are, I believe, hamster embryonic kidney cells. So these are mammalian cells. So we're not in a tardigrade anymore. These are animal cells. And so when you ask an animal cell to make the tardigrade protein, will the animal cells survive being dried out better? That's kind of the goal of this paper, right? Because if we have this magic protein that stops cells from drying out, can we make it somewhat relevant for human health in some way or another? And so to do that, they asked, genetically asked these kidney cells to make this tardigrade protein. And so on the left here, we see in blue the nucleus. So blue, we see DNA. That's what that blue is. And the green is I believe the cytoplasm. So like just the internal guts of the cell. And you can see that the guts here are pretty uniformly spread out. The protein that is being expressed is pretty uniform. It's not really structured in any way. It's just kind of all over the place, right? And then on the bottom here, we have the same thing. This is now a reporter that is linked to the tardigrade protein. So you attach a fluorescent molecule to the tardigrade protein, and when the kidney cells are happy and chilling, they look identical, right? I think we can probably agree that these look the same, right? Top and bottom image.
Yeah.
When we add sorbitol, so sorbitol is a molecule that can draw out water from cells. So this is their way of experimentally dehydrating the embryonic kidney cells. And now when we zoom in, how would you describe what you are seeing here?
What is going on at the bottom? I mean, it looks like it's exploding a little.
Yeah, so what we're seeing is the green fluorescence protein, in this case M Venus. What we're seeing is it's kind of aggregating in these patterns, right? So indicating that something is happening internally to shift the cytoplasm in a way where the proteins are now not uniform, that they're bundled up somehow. And so what the authors are saying is that this is the result of these tardigrade proteins forming these fibrous gels. So what you're seeing here is, it was uniform in the cytoplasm before, but when you dehydrate them, now they form these networks, you could call them. So I just thought that visually that was very interesting to look at. But the question remains, it forms a gel, but does it prevent the kidney cells from dehydrating? Do you have a question, Z, before I cut you off?
I mean, this is really cool. This is really cool. I mean, I had a brief question. Maybe I'm just not knowledgeable on how biology research works, but how did they make sure that the fluorescent molecule attaches specifically to the protein they want to look at and not to anything else?
Yeah, it's an excellent question. So you actually do this at the level of the genes. So you would, you can kind of think of it as just like typing code that adds the fluorescence molecule at the end of the protein that you want to make. So if you do that, then you're all golden, right? Because, central dogma, DNA to mRNA to protein, if you change the DNA, you can ensure, if you change the DNA of just the tardigrade gene, then you can add the protein just to the tardigrade gene.
Oh, I see. So you're specifically matching with the molecule, I guess, for the fluorescent molecule.
Yeah, yep.
Mm-hmm.
No,
this is really cool.
Marlon says, could you measure the amount of water remaining in the system after dehydration attempts? That is exactly what they are about to do in the data that I'm about to show. So Cas-D does not help to retain water more than other proteins or gels. You can see that the percent water content after desiccation is pretty much the same across the board. So we have here gelatin. So this is, it's gelatin. So this is a protein that forms a gel. And you can see that this gelling doesn't really seem to change the water retention by that much. Lysozyme, so this is just another protein. When you ask the kidney cells to make more of this, it also doesn't really seem to do anything. Cas-D, which is the protein that we're interested in, doesn't really do anything. And then I don't fully remember what FL Pro is here. I took a look at this paper this morning, so it was pretty last minute. But what is true here is that you can see that in terms of helping the cells survive desiccation, it really doesn't. It seems like Cas-D is not like by itself enough to prevent water from leaving the system, which is pretty mysterious.
Whoa. So that means our third hypothesis is not right, that it doesn't help to retain water more.
So the third hypothesis is looking in a bad way. It's looking not great. I hesitate to say that
Yeah.
it's false because the authors will explain here. So I'll just read this part out loud. The study does not rule out the possibility that gelation of the Cas proteins can be mechanistically linked to tardigrade and hydrobiosis, but it is clear that gelation of these proteins is not playing a role in water retention. So this is saying is that we know that the Cas proteins are important for tardigrade survival, like for sure. We know that when the tardigrade does not make this protein, it cannot survive. However, the gelation itself, like the forming of the gel, like that might be useful for some reason, but it might not be because it is retaining water. And this is also a flawed experiment as well, because just because this doesn't work in a kidney cell doesn't mean that it doesn't work in a tardigrade, right? It's two very different environments potentially. So there's a lot of I wouldn't give up on hypothesis 3 just yet, but it's not looking great right now. Put it that way.
Yeah.
So I'll continue reading. The formation of a gelled matrix of protein may serve as a desiccation-inducible cytoskeleton. These are very fancy words that I'll explain in just a second. That helps to maintain the organization and ultrastructure of drying cells, such that during drying, membranes do not collapse and fuse. Right? So I had mentioned earlier that when you have a system that draws out water from your cells, you get a collapse of those cells, and that can be deadly. So what this is saying is that maybe instead of retaining water, this is like a second skeleton for the cell that doesn't rely on the other cytoskeleton parts. So keeping the cell from collapsing in on itself, kind of like I guess, to relate to what you study, kind of like sinking into a singularity or something, like crumpling in a vacuum or something. Having something internal exerting pressure to make sure that the cell doesn't shrink and wither away.
Oh, I see.
Yeah.
But so I guess the cytoskeleton that's being referred to here, tardigrades have a cytoskeleton?
Yeah, so every one of your cells has a cytoskeleton. So the reason why your cells have a shape at all is because it kind of has bones. And those bones are proteins like actin and tubulin, these railways of proteins that help. They're kind of like, I would call them like support bars, support beams, right? Something like that, where they kind of dot around the cell and kind of hold up how the shape should be. The problem with the normal cytoskeleton, though, is if you remove the water, then they aggregate and get destroyed just like any other protein. But this tardigrade protein, instead of aggregating and being useless, actually aggregates into something that actually takes up a lot of volume, and that volume might help the cell from not collapsing in on itself.
I see.
Yeah.
That's really cool.
So I want to point out one more hypothesis here in the middle of the second paragraph. The sentence starts with liquid-liquid phase separation. So liquid-liquid phase separation of desiccation-related intrinsically disordered proteins, again, a lot of fancy language, don't worry too much about it, could serve to promote desiccation tolerance by sequestering and protecting vital proteins and other cellular components by setting up regions with chemistries, biophysical, or material properties that promote protection. So they're being very careful with their language here, right? But this is essentially pointing to some of your chatters and my chatters hypotheses that the floppy regions of that protein are providing a water-like environment, right? That's what they're saying with chemistries, biophysical, or material properties that promote protection. They're being even more careful than I am with calling it water-like, because I'm calling it water-like to make it easier to understand, but they're being more hands-off, just saying like, oh, maybe the gel itself is the environment, not necessarily because it has water, but because it does something that protects proteins from being destroyed. So very smart chatters, but we don't know if that's correct, right? This is ongoing research. So if you are a young biologist or a young biophysicist or a young biochemist, this is a legitimate field of inquiry. And I don't think there are a lot of people doing this kind of research, right? When I looked at the amount of literature on tardigrades, it's surprisingly not that well studied. So if you want to go into a tardigrade lab, you might be a part of a very exclusive group of researchers, which would be very fun.
And I heard that you can actually, like, purchase tardigrades as well. I mean, I'm sure there's a lot of regulations. You can't just, like, buy it on Amazon or anything, but
I mean, you might be able to.
maybe.
Because
I
they
don't know,
are
chat could
they
hmm.
are like very they're very common and they're everywhere. And also, as long as they're not like GMO tardigrades, I'm pretty sure the laws are actually pretty lax. But
Huh.
I'm not entirely sure. That would be my guess.
Box of tardigrades as a little snack, little
Uh
ton croissants.
if I ever get a sponsorship from a microscope company, maybe I do a tardigrade unboxing stream. That would be very fun.
Yes, that would be so cool.
That'd be really cool. She said it was cool. Guys, she said it was cool. Z's getting over her fear of tardigrades. It's happening.
Oh, I'm still scared, but I have a bit more admiration for tardigrades now. They still look creepy. I don't think I could ever look at them under a microscope, but
Toxo says
oh, now
it's.
chat is excited about this tardigrade microscope idea.
I'm pretty excited about it too. Toxo said it's only $17 on Amazon for a live tardigrade culture. That is very affordable.
What?
Yeah,
Oh my god.
as I was saying there, it's not like they're hard to grow, right? They live anywhere.
Well, okay, I'm just saying, I think it would be really cool if you did a tardigrade stream.
But I'm guessing
Wowzers,
you would
yeah.
not. I'm guessing you would not like to be invited to that.
I'll keep the video off, but I will be there auditorily. Tardigrades don't make
Stepwise.
sounds, right?
I mean, they're so small. How could you hear them?
Yeah, no.
All right. Okay, so this is why I have the slides say hypothesize too, right? So in my guide on how to never be wrong, if you're ever not sure about something, just say it's hypothesized. I think in my video I also say hypothesized because I wasn't entirely sure if anyone has proven it. So that's a note for anybody studying science is scientists love to call people out for being wrong, and if you never want to be wrong, just say it's hypothesized to do this, and then you're never
Mhm.
technically wrong. That's how you avoid spreading disinformation.
Hypothesized. Yeah, exactly.
Exactly. You got to be very careful when you are doing
Always
science.
hedge, yeah.
Always hedge, exactly. Hedge like a pro. Okay, so that was drying, and I'm not going to have time today to talk about all the other ways that the tardigrade can survive, but in order to kind of transition into understanding how this simple drying resistance can translate into other resistances, I'm going to take another look at something that you might find very relevant, Z, being the space girl that you are, is ionizing radiation, right? Something very important in space travel.
Mhm.
And so tardigrades can actually survive ionizing radiation, a good amount of it too, many more times than a human can. And so the question is, how
Mhm.
does this like drying vibe play into the tardigrade's evolutionary story? So this is what we're going to talk about next. So I have here just, gamma. I'm so bad. I'm so bad at physics. Every year that passes, I get worse at physics. A gamma photon. Z, help me out here.
It's isn't it just a gamma ray? Like,
Gamma ray. Okay, like I forgot the word for it. Sorry.
I mean, are you trying to just say that it's like a highly energized particle?
Yes, a highly energized particle.
Yeah. Well, this is definitely to scale. I'm just joking.
Don't call me out here, man. Not on my own stream. But yeah, so tardigrades can survive ionizing radiation, a very, very potent form of radiation exposure, which has all sorts of real, like, human health implications as, our ozone is thinning, as we are sending people into space, et cetera, et cetera. So it's a big deal that we try to understand better how to protect people from exposure to these elements, we'll say. So we'll take a look at how how ionizing radiation actually damages DNA so that we can appreciate how the tardigrade actually avoids dying to this. So there are two main mechanisms by which ionizing radiation can wreck DNA. The first is directly blasting DNA, right? So you can just have a high energy photon crash into DNA and cause it to break there. And if it doesn't repair correctly, you end up with a mutation. Mutations are bad because it can lead things to like cancer, right? If you get enough of this ionizing radiation damaging enough cells, that can lead to cancer. Although my audience can appreciate the mechanism by which DNA damage leads to cancer is debated, which I think is interesting to note. Another way that high energy ionizing radiation can damage DNA is indirectly. So direct is blasting DNA directly, and then indirect is actually crashing into water. Crashing into water causes it to become a superoxide or an O2 minus. This is a very deadly molecule, right? One of our greatest extinctions on planet Earth was because we had too much oxygen in the atmosphere. Oxygen is a tremendously dangerous molecule, and this tremendously dangerous molecule can actually do the same thing, destroy DNA, cause it to repair incorrectly, and then lead to deadly cancer. So these are bad.
Yeah, no. I mean, Xenotime's saying reactive oxygen species are scary but awesome. I think they're more on the scary end, but yeah, no, very, very scary.
Yeah, it's also why you hear these health influencers tell you all the time. Health influencers, also your doctors probably, tell you things like eating blueberries is probably good for you because it has things called antioxidants, which help clean up these reactive oxygen species. Okay, so the question then becomes, how do you think the tardigrade gets around DNA damage? And so I think this is, unlike my other questions, I think this one is more fair to hypothesize. I don't think that people will necessarily get the right answer, but I think there's a lot more creativity that could be true here. So how do you think the tardigrade gets around DNA damage? It's kind of a trick question, but it's kind of fun to just hypothesize, right?
I mean, like, I mean, it could probably, I mean, that protein that we talked about before is mostly for desiccation. Maybe it has some sort of other protein that can protect the DNA, or I have no idea actually.
Yeah, and I love to be in this space of I have no idea, because when I read papers, I'm often asking myself the same question, and it's always nice when I read the answer. I'm like, damn, Mother Nature is so clever. So we're gonna see how clever Mother Nature actually is in this case. Yeah. I do see the correct answer in my own chat, but I don't want to say it out loud again because spoiler reasons. Okay, so here are some hypotheses that have been thrown around. So we know that the tardigrade ton state can activate at low water, right? Dehydration causes the ton state, becomes a little croissant, and this ton state actually ejects water, right? So they are purposefully dehydrating themselves to protect themselves. This can be for a couple of different reasons, right? You can imagine that if you don't eject your water when you are stressed, then that water can be used to hurt you. The most obvious example is freezing, right? So if you freeze something, if you take too long to freeze something, well, what does freezing water do? Z, do you know kind of where I'm getting at?
Freezing water?
Yeah, if you took a normal kidney cell and you dunked it into not liquid nitrogen, that actually ruins the analogy I'm trying to draw here, but if you threw it in the freezer, do you think that kidney cell survives being thrown into the freezer?
No, I don't think so, right?
Yeah, but why do you think it dies? Or what is one reason why it dies when you thaw it?
I mean, I feel like if it freezes, I mean, the water kind of like connects together, right? It won't be like freely moving. I think another thing is maybe there's like not enough energy to conduct like basic things. The water could also expand and break things.
Yeah, there you go,
Ice
Z. There you
expands.
go. There you go, Z.
No, it's all thanks to chat. I was thinking about just freezing, not water expanding, but yeah.
Yeah, my chat's got it too. Explode, sharp crystals, exactly. So part of the tardigrade's self-protection function is when it gets too cold, it'll eject all the water, and it is hypothesized that is because if there's too much water around and it freezes, you get sharp crystals, you explode, right? Because water expands when it freezes. So, there's precedence for this. If you get rid of the water, there's less water that can expand and skewer the tardigrade from the inside out. It is tough being an organism of that size, right? And so a similar thing can be happening here, where if you eject water, there's less water around to be able to be turned into reactive oxygen species. And so you can protect the DNA a little bit by removing the water. And by removing the water, you can avoid the generation of these toxic compounds. So that's one way that the tardigrade can deal with DNA damage. The next way is shielding. And this is something that I came up with. I don't think anybody seriously thinks this, but my first thought was something like this. We know that tardigrade proteins aggregate, so maybe there's some kind of ultra protective protein structure that can block radiation, but I kind of realized on my own that this was kind of dumb, right? So
somebody in my chat did say that lead shield. Lead shield,
Yeah, that's why I realized that this was a dumb answer, and I don't want to embarrass myself in front of your audience because, well, I'm saying dumb as in like you are a physicist and know a lot more about radiation than I do, and I wanted to try to be as faithful to physics as I could. So if you didn't know that, then no shame on you. But I was preparing for a collab with Z, so I wanted to make sure I was correct. Lead and concrete are used to block radiation, but lead has a very large nucleus, right? It has a large surface that can actually stop radiation from coming in, and the molecules that make up proteins like carbon, nitrogen, hydrogen, oxygen, sulfur, phosphate, they're tiny nuclear wise, right? They don't have the bulk needed to block an incoming photon. So I thought it was a cool idea, but then I was like, don't want to embarrass myself in front of Z's physics audience.
Yeah, and I mean, if you're thinking of highly energized particles, one lead atom wouldn't be able to stop that. You would need a lot of it, and it would have to be kind of thick too, if you think about it too. So
that's true, yeah, because you need layers and layers to make sure that it will hit a nucleus before it passes through.
Yeah, atoms are mostly empty space, so
so I quickly realized this was not an idea I should be saying seriously. And then there's hypothesis 3, DNA repair. So someone in my chat pointed this out, that tardigrades might have a super crazy ability to repair DNA. And I'll just say as a spoiler that they absolutely do. They have very unique ways to repair DNA, and we're actually going to see how that ties in with this desiccation story. So not only do tardigrades have an amazing capacity to repair DNA, but anyone who's come to any of my cancer streams know that you also have an amazing capacity to repair damaged DNA. So if you break DNA, if you break only one of those strands, the non-damaged strand can act as a template for the other strand to repair correctly. So even though you have a lot of exposure to things that could damage your DNA, you go out in the sun and you don't wear sunscreen, you just exist, you get stressed, all these things can build up these these reactive oxygen species that can damage your DNA. But you get to live because you have the capacity to repair that damage. But if you were exposed to cosmic rays, your repair could not keep up with the damage, right? Your DNA would get shredded, and that would be very bad for you. A tardigrade, however, can repair DNA after exposure to radiation, and it was recently discovered, very, very recently, I think it was just last year, actually a couple months ago, a new family of proteins. This is actually so new that I don't even know what the protein looks like. They don't even have a structure for this thing yet. It's called TRID1.
Whoa.
So yeah, there's no graphic here because there's just, it's that brand new, which is why I'm encouraging anybody who loves tardigrades and is a biology student to, maybe get in on this, find out where these people are, and try to do their science. So this is called tardigrade specific radiation induced intrinsically disordered protein. So this is a very descriptive title. So it is, so this protein is intrinsically disordered, so this already gives you a hint. This is an aggregating protein that reacts to radiation. So how does that happen? So the way that this happens is that, so there's no cartoon of this available, so I drew one messily in Photoshop, is that TRID1 actually aggregates into these pseudo bubbles, which encapsulates DNA repair proteins on the inside. So
What?
biology, yeah, I know, right? Pretty insane. So there must be some part of the TRID1 protein that binds to DNA repair proteins or binds an adapter to a DNA repair protein. I don't know which one, and I don't think they know which one, but they found concentrated DNA repair protein within these protein bubbles that then get recruited to broken sites in the DNA, which actually enables them to fix DNA to keep up the pace with the damage done by ionizing radiation.
Holy smokes, this is crazy. Like, how did evolution even think of this?
Yeah, and that's the thing that we're going to talk about next. I wanted to cover radiation because of your interest in space, but
Yeah.
for me, this is like there's no way, because the tardigrade didn't evolve this pressured by ionizing radiation, right? That's just not a thing. We have an atmosphere.
Yeah.
Right? Like the tardigrade didn't evolve to survive cosmic radiation. It just does this by accident, kind of. Asterisk, right? Like I don't have the complete evolutionary story of the tardigrade, but there's almost no reason why the tardigrade should have all these superpowers unless there was some shared mechanism by which the tardigrade could develop this strategy. I think that's really cool.
Wow.
This is not the only DNA repair pathway in tardigrades. It's just the one that I found to be the most compelling to tell with the time that we have today.
Mhm.
So here is my idea, right? I don't know if this is true, so this is all hearsay. This is my hypothesis. I am guessing. This is the general trend of what happens when anything alive experiences getting dried out. Desiccation leads to protein aggregation and also cell death, and cell death plus protein aggregation is just straight up death, right? So this is protein aggregation. I'm going to call that a lemon, right? I'm going to call that a lemon. The thing about the tardigrade, though, is that through eons of evolutionary pressure, I think the tardigrade evolved an adaptation to make the most out of a bad situation. I think because the tardigrade experienced desiccation so frequently, it evolved a set of proteins that still does something when aggregated. So the tardigrade's lived reality is, I'm going to encounter hardship, so I'm going to need a protein that can deal with hardship. And because the tardigrade is always being dried out and rehydrated, it figured out, again, quote unquote figured out, Mother Nature figured out a way to turn this lemon, this sour, unappetizing aspect of protein biology into something that is actually very useful, lemonade. So instead of dying, the tardigrade harnessed the power of aggregation to actually survive. And I think that's amazing.
Wow. Yeah, because I was gonna say, like, we have an atmosphere that protects us from radiation. Radiation is not a threat that these tardigrades are dealing with on a daily basis compared to like desiccation, which could basically happen at any moment on a leaf, right?
Yeah, and there's no way that tardigrades evolved to survive getting plunged into liquid nitrogen, right? There's just no way. There's not a place on planet Earth that reaches that temperature except in physics labs and in biology labs, chemistry labs, right? They didn't evolve in liquid nitrogen. The coldest it gets on the planet is nowhere near liquid nitrogen temperature. So how the hell did the tardigrade evolve to do that? In my hypothesis is because this principle of, if we're going to get wrecked anyway, we need to be adaptable. I compared this to like spiritual philosophy, in my video of like, if you're very zen about things, if you're like go with the flow kind of evolutionary strategy where you accept that you are going to experience some kind of physical, chemical hardship, then it is maybe better to adapt to that hardship than it is to fight that hardship, right? Because you can imagine that an animal has two strategies: either just avoid ever being out of water, or accept that you will be out of water and evolve something crazy to deal with that.
Mhm. Wow.
Yeah. So I'd say that's pretty good for an animal, right? It's not a surprise that an organism can do this, right? Because bacteria do this all the time. It's not that crazy. Yeast can do this. It's not that crazy. Archaea are famously very happy to be in boiling water, very happy to be in very salty conditions because they evolved to do it. But the thing is, it's a lot harder to keep a multicellular animal alive than it is to keep a single-celled animal, not a single-celled animal, a single-celled organism alive.
Mhm.
Bacteria can sporulate, yeast can sporulate, they can form really hardy uh, uh, structures that can allow them to survive all sorts of things. But for an animal to do it, like a real animal, given it's a very small animal, but this is still an animal, I just find that is really, really incredible. Um,
Wow.
yeah. I think I have one more slide here, and that is
Mhm.
looking forward into the future. So I didn't have time to throw this in like a very detailed way, but I wanted to share this paper title called "Properties of a Tardigrade Desiccation Tolerance Protein Aerogel," which I figured would be of interest to you, Z, given your
Huh.
background, where like scientists, materials scientists, are trying to play with the properties of tardigrade proteins to do things like make aerogels as well as dry vaccines. So part of what made COVID quite difficult was this idea that we needed to ship out the COVID vaccine in minus 80-degree freezers, right? And not every hospital has a minus 80. It's also very expensive to maintain a
Mhm.
minus 80 because if you had the vaccine at room temperature, it would degrade. So scientists are thinking of using the tardigrade protein, this protein that can act kind of like water without really being water and can help proteins survive a dry environment, to preserve vaccines in order to keep them around for longer so that patients can get treatment for cheaper, right? And I think that's really incredible that this like little goofy critter that the internet loves is actually really an interesting organism to study for biology, for physics. And I think that's lovely.
No, this is so cool. I mean, it's basically using the material, the physics properties of the protein for human benefit, right?
Yeah, yeah, exactly. Like figuring out how to make, like, I don't know anything about aerogels, by the way, Z. I don't know how much you know about them. I just, it looked
A
like a physics word,
little
so I put it
bit.
on there. But they
Yeah.
do have unique chemical properties that allow them to be used in all sorts of very interesting ways that I never would have known if I didn't do my video.
Wow. I'm gonna be reading this one right after the stream. That's so cool. I mean, interdisciplinary connections, right? So that's so amazing.
Yeah, I actually really love the opportunity to do this collab with you, Z, because it forces me to think outside of like my biology uh, echo chamber, right? To think about how the science that I love so dearly can be applied to the science that other people love so dearly. Um, and yeah, I think it's really neat that this little guy ended up being like the collab that I think we really should have done. Because yeah, we talked a little
Yeah.
bit earlier about how like we had a lot of other ideas that didn't really like hit the same.
Yeah, no, this was great. And I also wanted to say, like, thank you so much for making this presentation incredibly accessible. As somebody who is I would say, like, I don't, like, suck at biology, but, like, I'm not the best at biology. I haven't really thought about biology in a long time, and this really was it was really accessible and was also pretty cool, and now I think, yeah, tardigrades still look scary, but honestly, the ways you can use the tardigrades now, I'm a huge fan of that.
Yeah, I don't think I could have ever realistically convinced you that the tardigrade isn't a little horrifying. It's like the cutest eldritch horror that you can experience, right? Like, it's cute, but you have to admit that it's kind of horrible too.
Yeah.
But it's the cutest version of horror I think that is out there. But in terms of like, I think there's a lot to respect about the tardigrade story. Like, not only can it really take a beating, but also it comes from such humble origins, right? Just on a leaf, hanging out, getting dried out. None of us, you know, who amongst us hasn't bought a humidifier for when it gets cold? But to a tardigrade, that drying out would be deadly. And to adapt to that dry condition, and as a like, in my opinion, right, someone might prove me wrong later, but from my perspective, that humble environment allowed the tardigrade to accidentally get all these other superpowers as well, and I think that's a very interesting story.
Yeah, no, this is absolutely spectacular. Chad is asking tardigrade dating sim what? Okay, I don't think I'm at the point where I love tardigrades that much, but I actually do know a few people who are actually working with tardigrades. mostly just for experimentation, not really for research purposes. Um, but now that I have a little bit more confidence in tardigrades, maybe I'll be able to see what they're doing instead of running away and screaming.
I think we called it a W, chat.
I
I think we
would
called it a
say
dub.
I have positive mindset towards tardigrades now, which is a little bit better than where I was before. Yeah,
Yeah.
W in the chat. W in the chat. Woo tardigrades, exactly.
All right. So, yeah, because we have a guest on, I had been mostly ignoring chat, but now I think we can take questions or comments from both chats. So if you have a comment that you would like for me to respond to, I'd be happy to answer that now. Yeah, sorry for not getting to those, but I just felt like having a conversation with my fellow V Theorem member Z, and getting to share something that I find to be so lovely about yeah, the molecular nature of life, I should call it. It's interesting to note as like questions come in to talk about some of the other collab ideas that we had. Do we want to share those, or do we want to keep those secret?
I mean, we could probably share those. I mean, we had some really cool ideas. I think my favorite one was we were thinking about doing a collab on how eels electrocute. And there was like a circuit looking diagram with the eel and like biting into a human. That was one of the ideas we had.
Yeah, and the reason why I think like I don't think that's very good is because or it's a cool idea. I just don't know what my angle would be because I don't know how to do EM. I haven't done V equals IR in many moons.
Oh no. Yeah.
To be honest, can I be honest? I still kind of am not confident that I know what the concept of voltage is.
Oh, like, yeah. I mean, I think it took me quite a while to really understand, but then I saw a video once that was like describing voltage as like water moving down a hill, and you kind of think of it like potential energy because
Yeah.
voltage is electric potential. And so you can actually visualize it as like water flowing down. So the water, or in your case, like electric current, goes from like a place of higher potential to lower potential. And that was really the way that I kind of understood it.
I
Um,
I think what would be really important for the eel collab, which I think it'd be really fun, is I don't understand what part of electricity kills you. Is it high voltage? Is it high current? Can you die from high voltage, low current? Can you die from high current, low voltage? Like that part, I don't fully understand because I think you have to know how electricity travels through the body to actually understand that. I wonder if anybody in chat has some kind of insight as to how those work because it just made my head hurt.
I believe it's high voltage. No, maybe. I do have some friends who work with like high voltage machinery who will know a lot more about this than I do. I haven't thought about this in quite some time.
Yeah, because
Is it current?
when you walk by like workplaces, you often see like warning high voltage, but I'm like, is that the thing that kills you, or is it high current that kills you?
Well,
Am
I think they're kind of related because if you're dealing with these incredibly high numbers, like the resistance of the body is not, I mean, I guess depending, I don't think it's that high. Yeah, there's different routes through which it can kill you. Yeah, but yeah, this, I think this comment describes it quite well. Current kills you, but you need voltage to create current. So they're kind of like hand in hand with each other.
Leo says there's an excellent styropyro video on this. Oh, I might give that a
Ooh.
watch because reading like Wikipedia on V equals IR did not like did not help. I tried a good amount. I've been trying to do this in some form or another since like 2 years ago, and I just never learned enough EM to like be confident enough to talk about eels on stream. Although that's something that I definitely want to do one day.
Well, for the record, I feel like I'm also not as like I haven't really thought about like, well, there's magnetic fields out in space, right? There's interstellar magnetic fields, but that's a very different behavior to, like a magnet on Earth or a current going through an eel. So, yeah, I think this tardigrades idea was really great.
Yeah, because it's basically pseudo astrobiology. I don't think it's like real astrobiology in the sense where the tardigrade is not an extraterrestrial organism and it's not like hypothesized to be an extraterrestrial organism. Because again, you bring the tardigrade to space, it's going to be in a ton state. It can't actually survive out there for very long. But in terms of like the perfect intersection, because we had other ideas where we were talking about like forces, like Newton kind of stuff. But it's like I was talking to Mylan about this, or Mylan, I don't know, I forget who it was I was talking about, this idea of like, we are usually so siloed in our own academic specialties where we're like pretty ignorant of what everyone else is studying. So anytime I meet a physicist, I'm like, you're good at EM, right? You know all the Newton things and you know all the electromagnetism and quantum things. That's a huge assumption, because anybody who comes to me and goes, oh, like, do you know animals? I'd be like, no. I don't know anything about animals.
Yeah, because I think, like, I mean, at some point, like, everybody has it on the way to become like a physicist, right? Like, you have to study like ENM, you have to study quantum. But as you sort of, focus more on your preferred direction, you sort of like there's a graph that shows this. It's like at first it starts broad, and then your knowledge sort of like goes to a peak, and the amount of things that you know like becomes very narrow, but you know a lot about it. um and so I think that's sort of what it is, right? Like, at some point, yes, like, physics people like have studied E&M, do know physics, can probably solve like a V equals IR equation, though. Yeah. Griffith's my love. Yeah, no, there's this textbook that's out there that's really great for it. Um,
uh,
so yeah.
yeah, I mean, that makes a lot of sense. And again, yeah, if anyone asked me, yeah, to name things about animals, I would just lose my mind. My chat is saying that it's high current, and that is what I read online. That it's consistent. I just want to acknowledge that my chat has answered me. Thank you for that,
I
chat.
think the I I think there's a comment here that said high voltage is what can basically stop your heart, and then high current is what burns you internally.
Oh, such a headache.
Yeah, no, I
It's
haven't
such a
really thought
headache.
about this either now that I think about it.
Yeah, I need to know. I need to understand from the ground, because like I'm taking your word, chat, but like I need a fundamental in order to do the collab or the video. I haven't decided entirely yet, but in order to do that content, I would need a ground up, confident understanding on the basic mechanisms. And I even through your answers, I still have to review. Like, I need a video. I'll watch the video that Leo had recommended. Hey, Leo, if you're still around, if you want to post that in the Discord, that might actually be very helpful.
Yeah, the Styor Pyro video, I'll
Yeah,
definitely need to
yeah,
check it I
yeah,
think my phone is going off. Give me 1 second.
yeah. No worries. Yeah. I saw some other. Yeah. To address Rico's comments, he doesn't love tardigrades. Z finds them to be like eldritch horror abominations, which you gotta be understanding. They are eldritch horror abominations, but they are cute eldritch horror abominations. I think that is the centrist position in the tardigrade debate. Yeah. You can't really say voltage or current because voltage is necessary to push current, but many high voltage supplies can't supply much current either. Yeah, that's the part that's difficult for, like, I would need to know from the biology perspective exactly what it is that causes cell death. Like, yeah, just like how current moves through the body and how that affects biology, to really be able to understand, because that eel paper has like math in it and I want to do something with that math, but without having a confident understanding on how circuit diagrams work, I'm not willing to show my whole ass in front of YouTube.
Yeah, no, I feel the same way. Like, I feel like circuit diagrams can be a little hard to deal with. And I feel like something like with an animal, you don't I mean, like, what's the resistance of an eel, right? Like,
Yeah, that's like
feel like
another question that I was like, okay, so in the circuit diagram, they're showing that the resistance travels back down the eel. That this is so funny talking about a paper that I can't even show on stream right now. But the idea is that like there are multiple routes that the electricity flows through that are available to the eel, and the idea is that the jumping height of the eel is actually correlated with how much shock, whatever you want to call it, current, voltage, whatever. No, the voltage is intrinsic to the eel because it has an electric organ. The current changes due to the height that the eel jumps because it changes its resistance. And it's just like, oh god, I haven't had to think about this in years. The way we do physics. But
Yeah, I feel like if we had went with that, it would have been a lot more physics focused. I think what's really cool about tardigrades is that yes, there's a lot of biology that's taking place, but there's also a lot of space slash like physics implications, right? Like one of the things we're trying to understand is like I actually just recently listened to a podcast about trying to have people hibernate um and trying to protect them from radiation. And one of the things that they're studying is like they briefly mentioned like tardigrades in the ton state, but the main focus of the podcast was like understanding hibernation in animals and seeing that they actually become more like radiation resistant and trying to bring those same applications to potential human habit hibernation as we go like to Mars or to even further destinations. So I think it's really cool in like seeing the applications of tardigrades in space.
that's really interesting. So these animals get more radiation resistant or more mutation resistant?
More, I guess more mutation resistant is probably, you're right, the better word for it. I'll probably have to go back and I I'm going based off of memory right now, but they were basically able to bring different animals and put them in a hibernation state to the point where they were using like 0.01 calories a day or something. It was crazy. So they had gotten their metabolism down like so low that this could be applicable for space travel, for instance.
I think that as a biologist sounds a little more correct than radiation tolerance in hibernation, because I can actually imagine why that would be true. Z, do you know who the biggest culprit is in damaging your DNA? Just like sitting
Oh,
around enjoying a meal, whatever, your DNA is being damaged all the time. What do you think is causing it?
I mean, rate. I mean, I don't think we don't have that with
Assuming you're
radiation, but
indoors and protected from the
I'm
sun.
doing indoors. I
Yeah.
don't know, like, would there be, like is it, like, the free radical oxygen or something, or
Yeah, free radical oxygen produced by, drumroll please, your powerhouse of the cell, the mitochondria. So if you're reducing your
Oh!
metabolism to like zero point 0.1 calorie a day or whatever, it's probably because your mitochondria aren't ruining your DNA, right? Like biology is a very complicated thing that oftentimes doesn't make a lot of sense. But as a byproduct, your mitochondria are making, as the
Huh.
final electron acceptor, water is so sorry, let me make sure I get this right. And someone can correct me if I'm wrong, but in the aerobic respiration pathway, you get the shuttling of electrons, and the final acceptor of that electron is water. And the thing is that once that water gets hit with that electron, it can temporarily form hydrogen peroxide, which is one of these free radical producing molecules that's bad for your DNA, and it's doing it all the time. The reason why it doesn't kill you is because you're repairing your DNA all the time. And there are things like
Hmm.
antioxidants that take away that oxygen. So I actually do believe that that's a genuinely interesting way to protect people in space, is to induce a level like a biological hibernation so that they have less active mitochondria, would be my guess.
Wow. So the mitochondria is the killer of the cell and the powerhouse of the cell, I guess?
We have a complicated relationship with the mitochondria, and actually there's been a lot of interesting discussion in my Discord about the role of mitochondria, how they evolved, and is it really as amicable as we once thought, or are we always in like miniature combat? I've not read any of the papers, but I have been reading the conversation around that, which I think is really interesting. Oh, sorry, thank
Yeah.
you. It's so useful having Lunkel in chat right now. Terminal acceptor is oxygen. The normal result is water. The bad version is hydrogen peroxide. Thank you. That's an embarrassing error to make, honestly. That's a bad mistake to make. Thank you so much, Loco. Yes, the terminal acceptor is oxygen. That's why it's oxidative. Sorry, yeah, it all just
Hmm.
gets jumbled in my brain after not reviewing it for a while.
Yeah. But this is so cool though. I might actually need to head out soon, unfortunately.
Yeah, no problem. Uh,
But I
Z,
did want to say,
yeah.
um, thank you so much. We finally made this collab happen, and this is actually our first collab ever. Um, and especially now that I'm part of Ethereum. So yeah, this has been great.
And can I say something, Z? I am now,
Yeah.
I believe, the first person to have collabed with every other member, or did you as well? I can't remember.
No, I haven't yet. I
Yeah,
haven't yet.
I think you were the last person that I have yet to make contact with directly. So I
Oh,
won. I've crossed the finish line.
you defeated you defeated V Theorem. Now we need to debut a new generation.
Let's not talk about that. I'm not ready.
Gen 1.618 when.
When indeed.
Well, this is great. Yeah. I mean, speaking of collabing with V Theorem, a brief announcement is that next Tuesday I'll probably be streaming something. I think I'll probably be going over some astrophysics papers since I've been delaying that for so long. And we'll be dropping my next video in the partial differential equations series. Um, so I'm super excited to share that. And then this next Saturday, I'm actually collabing with NEP for the first time. So I'm super excited for that as well. We're gonna be um just asking some basically doing a free Q&A with NEP and asking some questions on horticulture. We had a really great conversation, although it was very brief on like the LA wildfires during the charity fundraiser. So it's basically going to be like a chill continuation of that. So yeah, that's just some brief announcements from me.
Yeah, and your PDE series has been doing numbers. I'm honestly so impressed. It's very exciting to see the algorithm.
Well,
uh,
oh, thank you, Shiraho, for subscribing. But yeah, no, the series is really taking off. I think in like 24 hours, it was actually the most viewed video on my channel. So, and all the kind words and the questions in the comments have been great as well.
Oh, that's awesome. All right, everyone, go give Z a follow if you do not follow her already. Thank you so much for taking the time to chat with me on this channel and allowing me to soapbox about one of my favorite microorganisms. And I wish you good luck in your future endeavors. Looking forward to the collab that you have going on with NEP. Yeah,
Yeah,
and
and
we'll chat.
I think a lot of Orbeez are probably subscribed to both channels, but if you haven't already, go subscribe to Phi. I think I linked his POV in the pinned comment. So if you go there, you can just click subscribe on the channel and go check out the Tardigrades video as well. I'll be doing that later today as well. But that's all from me. Thank you so much for collabing, and we got this out of the chat. So yeah.
Yeah, I'll stick around to hang out and chat with my own chat. But thank you so much for coming by, Z. I will talk to you later. Bye bye.
All right, goodbye. And Orbeez, I will see you guys next Tuesday. So goodbyes. I'll see you in the next one. Bye bye. Bye bye.
Bye, Z.
Goodbye. I'll also leave the Discord call as well. Let me
Yep.
do that right now.
All right, yeah. So I'll stick around and hang out and chat just a couple minutes before I take off, just because I feel like I've been wanting to stream, but I've also been stuck doing this flu video, which I said was going to come out this weekend, and I should have known better because the flu is always more complicated than I expect. But it's so miserable. Not to set the like tone badly, but it's just like the flu every time I see it come up in a headline or something, I have to revise my script or make sure that everything is still correct because like the stakes are what's really making it very stressful for me because I want to really make sure that everything I get is completely correct. But it is in its final stages because I am now editing a part about exciting new universal flu vaccines, which I think you will all enjoy. I've learned a lot. I've learned a lot about vaccines having to study for the flu because I don't know, the way that you learn about vaccines in school is like very minimal, right? Like you get live or attenuated viruses, you inject those, and then your body fights it off. And that's kind of all the detail you get. And for a long time, that was like fine for me. Like I didn't really bother questioning all that much. Would you say that this weekend kind of flew by? If I need your flu video to educate my immune system, I've been really sick for a week. I would also like the flu video to come out, but I just like got the whole vaccine part wrong in my first couple of drafts of my script, and now I finally have come to a place where I believe I understand what the hubbub is about, and it unraveled like a huge rabbit hole that I've been unable to escape from, because the idea that you just inject a little thing and it trains your immune system, that's correct, and that explanation works for, I'd say, 99.9% of people on this planet. Like, you don't have to know how a vaccine works beyond that. But when it comes to designing a new flu vaccine, the actual details of how your immune system trains all of a sudden becomes very, very important. And I was just like, I should have taken an immunology class when I was in school, dude. This stuff is so complicated. There's like a localized response effect to immune training. There are a couple of different immune modules that exist all at the same time. Different cells get trained in different ways. Different cells, like different immune cells, take their trainings in different ways. And that's kind of the part that I'm struggling to get through when it comes to a universal flu vaccine. Because as much as I doom about the flu in public, reading the vaccine literature is actually very, very exciting and hopeful, right? Like if you've been dooming about bird flu, you're not wrong to, but there's also reasons to have hope. And I want to make sure to strike the balance of like, I don't want to send people into a death spiral. I also don't want people to underestimate how bad the situation is. So yeah, just it's going to take a bit longer, but I wanted to tease just a little bit that like, we have to take the stupid flu shot every year because our current meta of the flu vaccine is just like not that, it needs updating. It's like not been that different since decades ago, right? But there's new stuff coming out that I'm very excited to show you that I just want to get it done. Hopefully by the end of next week, by the very latest in 2 weeks, I will have the full thing done, and I will be very glad to finally get it off my chest. But I kind of wanted to share like one, I think, wholesome story from last week that I think would be just nice to talk about on stream, because it's just a nice thing to share and leave you with a bit of positivity here. The slide here reminded me of Cave Johnson's Lemon Rant, and I have no idea why. I don't actually know what that's referring to reveal it. But yeah, so this wholesome story. So I have come to appreciate that like my current class that I'm teaching actually is like very, very good. Like in spite of insert current events, I actually am realizing that like the current batch of students that I have are incredibly motivated to learn, and they're actively very hungry to be the best biologist that they can be, to the point where like I'm a little ashamed of myself. I'm like I remember being their age and not being nearly as motivated, and to see like a generation of young people come up as scientists being like this hungry for, what I assume is like a dream career for them, was extremely, extremely nice to see, I'd say. I describe their attitude as sort of dogged, in a way where like a dog, if it is motivated enough, will bite down and won't let go until it wants to, or you can't shake the dog off. And I feel that way for some of my students, where they will not let an issue go without fully understanding what is behind the scenes. Whereas when I was their age, I took a lot of simplified explanations for granted. I was just like, oh, okay, so if that's what the textbook says, that's what the textbook says, and I just have to memorize that and accept that as truth. But I have not seen in quite some time students who have been this motivated to have a deep understanding of how, the molecular nature of life. And and I find myself really racking my brain, like very challenged by them, which it's not unexpected, but the fact that so many of them are challenging me either means that I'm getting not as cognitively quick, or it's because they are a special class, and I find that's really wholesome. It's great. It's extremely rare to hear that from instructors at any level now. Yeah, I mean, to be honest, like I felt, I don't really talk about the negative aspects of my job all that much, but there was a sense that, like, there's a worrying trend in at least American education, and I think it's because I haven't been teaching for that long that, in my career, I've not seen that many generations of students. So any trends that I could pull out are done from a very small sample size. But what I will say is that, like, from the general vibe of the situation, I think this class that I'm currently teaching is sort of like, I don't know, I'm very proud of them. And I think that is worth mentioning. Not because I think any of them will actually hear this, tucked away in this Tardigrade stream, but it's worth sharing, I think, a perspective from a current teacher in all of the science madness that we find ourselves in, that, like, they are very motivated, and that's awesome. I wonder if the ability to look things up on the spot changes how one interacts with taught information. I'm curious as to what you mean by that, or how you think it interacts with taught information. And like the questions that they ask me are not easily Googleable, which I think is an important distinction, right? Because they're if you go in places where, like, instructors hang out, there's this trend of a loss of self-efficacy, this idea of students becoming more dependent on their teachers for even the most basic of information. But I think that, there are fluctuations in, like, classes that might not have anything to do with anything, right? Nothing about, Gen Z is all cooked or whatever. It's more like sometimes you get students who are more reliant on you as a source of knowledge, and some students who aren't as reliant on you as a source of knowledge. And what I find is that the questions that I get asked nowadays are ones that it really makes you think. It really makes you think in interesting, complicated ways. And I end up having to start a lot of my answers with, I'm not sure, but here is my hypothesis. And I think those are the most interesting questions to get asked. Whereas, like, the adage of there are no stupid questions, that is true up to a degree, but if it is a question that you can Google and get an answer in 15 seconds, then it's not a stupid question. It's just a question that could have been addressed on your own. And I want to make that distinction, because I think interesting questions are like, it genuinely makes me very excited to see where some of these students end up, and I think that's wonderful. So yeah, I just wanted to, in the last few minutes of this collab, just leave you with something nice that occurred to me in the past week of my life. Yeah. All right. I hope you all enjoyed hearing about tardigrades. And I'll hang around in case anyone else wants to bring any other conversation topic up. But otherwise, I will end the stream soon, unless there are last minute things that y'all want to chat about. Love it when a student asks questions like that. Yeah, I tried to do that as an undergraduate for a very, like, not cynical reason, but, like, I made it a point as an undergraduate to always ask one question after class so I didn't hold up the class. And I would really try to make it an interesting question. Not like a got you question or anything, but, like, I wanted to challenge myself by seeing whether or not the answer that the professor would provide was one that they had to pull from their broader knowledge for, just to see if I could start coming up with my own new ideas, right? So not to, like, be like, oh, I knew more than the professor, but, like, if I identify something the professor doesn't know, it means that I have the capability to have a new idea. because ultimately the greatest insecurity I think for up and coming researchers, like undergraduates, is this idea of like everything's already been done. What the heck do I have to do? And I realized that the only way I could actually get to a point where I can identify a new thing is if I try to push the limits of someone who is much more senior to me. And I think the fact that, well, I'm not a professor, so I don't have decades of biology experience, but the fact that these undergraduates are already pushing my knowledge shows that they are pretty intellectually mature, and that is really exciting to see. Like in the past, if you were taught something that you might not be able to find other ways to reason about, the taught concept might expose new ways to unravel it. If you're taught something you might be able to find other ways to reason. I'm sorry, my brain's not working fast enough to comprehend. Taught something you might not be able to find other ways to reason about. To find the border of current knowledge. Yeah, that's what I'm talking about. Like, it's really hard. Anyone who's written a project proposal knows it's really hard to find a new idea, especially because like it's especially relevant because in my class the students, it'll be a couple weeks, but then they will have to find a new idea in biology to investigate, and that's always, I think, such a fun period of time, because for some students it can be very traumatic. Like some students take it very poorly, right? You ask them to come up with a new idea in biology and they do not have a fun time trying, and that's informative, right? That might inform you that maybe a research career is not for you, right? Because a research career is all about finding new questions to solve, new important useful questions to address, and so the fact that this part of this semester is coming up where students are going to be trying to find a new idea is really exciting, and they're doing it at a much younger age than I was doing it at, because in my education they waited until my senior year for us to even make the attempt to try something new, whereas they are getting it in their second year of college. So these are very young college students, underclassmen, that are being asked to find the limit of biology. And I think that they're all hungry and passionate enough to do it. And I think it's going to be very rewarding to see what comes out of that. Other points of view, alternate models or metaphors. Yeah, any dimensional thinking can be very, very helpful. And being able to attack a problem from multiple angles and not be satisfied with what you're fed is, I think, a mark of someone who would do well in like a research heavy environment. I was gonna say, yeah. I had a piece of feedback from a student who had said that, like, for the first time they had felt like when they were reading a paper it finally started to make sense to them, and I was like, that's such a good moment, right? That's such a lovely. I'm very fortunate to have a job where I can experience the moment where a student can be like, this gobbledygook, this academic gobbledygook. I do journal clubs on my stream, but I tend to cut out a lot of the text. Like if you're not in biology or don't have academic background, the first time you read a big boy paper, I guess I can't speak for everybody because I don't know outside of my discipline how difficult it is, but in biology it's very difficult. Everything's a freaking acronym. There are all these vocabulary words you've never heard of, all these different ideas that you've never seen, and you're expected to comprehend and critically judge what they mean. And yeah, I've told the story before, but it took me hours and I didn't even finish my first big boy paper, and I winged it when I had to present it. But the first time a student is empowered in a way where they go, oh, I think I could do this, is genuinely very, very heartful, right? It's like empowering, and it reminds me of Blue Period, where it's like he's beginning to believe, like that whole idea. And I get to, I've been able to experience that every semester, and it's about to happen again, and I'm excited for it to happen. Sometimes if I can't sleep, I ask Phi to read me random Wikipedia articles. That is true. I have done so. I have been told that my voice is very good at that. I've also noticed students sleeping in my class too, so I mean, while it might be beneficial if I wanted to take a nosedive into ASMR content, not exactly conducive to active learning in a class environment. I totally get it though. Like I slept in classes too, so when students sleep in my class, I'm actually not offended at all because I'm like, I was also in your shoes. I don't react very well to coffee. Like when I have coffee, I feel terrible, so I don't have the option of having caffeine to stay awake. I just have to work hard later, and I did fine in my classes, despite falling asleep in class. It just meant that the work I had to do after class was over was a lot more stressful than I think it needed to be. All right, I am done yapping here. Yeah, again, thank you all for coming to my little Tardigrade stream. It was fun to revisit an old Find the Neutral video and take a look at it. And if you've watched that Tardigrade video, I've hoped I've satisfied a bit of your curiosity to see where that Tardigrade research has gone. And I don't really know when I will stream next. It might not be next week because this week is really busy for me, but I really, really, really hope I get this Tardigrade video out at some point. If you haven't joined the Discord and you want to continue the conversation afterwards, you can go ahead, type !Discord. And with that being said, I will see you all next time, whenever that might be. Bye, y'all. Bye-bye. Bye-bye.